name is Emilia Natasha binti Sayhani from Bachelor of Science in Biomolecular Science AS244. My final year project title is Molecular Identification of Processed Tuna in Markets around Klang Valley. Fish is a major source of protein in Malaysia, and tuna is the one of the most popular and commercially important fish in the market. Different tuna species have different market values, taste, and quality, as well as conservation status. Some species are more valuable, while others are at risk due to overfishing. Tuna is sold in both fresh and processed forms such as fillet, canned products, and fish balls. However, processing removes important morphological features like the skin and fins, and this making visual species identification very difficult. Therefore, identifying tuna species based on appearance alone is unreliable, especially for processed products. Processed tuna products increase the risk of species mislabeling because the original physical features of the fish are no longer visible. Mislabeling can lead to consumer deception and economic fraud, especially when lower value species are sold as expensive tuna. Klang Valley was selected for this study because it is a major urban area with a wide distribution of tuna products. About 16.2% of the population in Selangor consume fish, including tuna, that indicating high market availability. To address this issue, DNA barcoding uses the mitochondrial CO1 gene was applied as it allows accurate species identification. This study used a CO1 PCR-based approach to verify the labeling of tuna products sold in the Klang Valley. This study has three main objectives. First, to extract DNA from tuna species samples collected from selected wet markets and supermarkets in Klang Valley. Second, to amplify the CO1 gene region using PCR technique for species identification. And lastly, to assess the accuracy of product labeling to detect any possible mislabeling among the samples. This study began with sample collection of tuna from selected wet markets and supermarkets from 5 different locations in Klang Valley. Next, DNA extraction was carried out and the extracted DNA was assessed for quality and purity to ensure it was suitable for downstream application. The mitochondrial CO1 gene region was then amplified using PCR techniques with 3 different primers to improve species identification reliability. The PCR products were visualized using agarose gel electrophoresis to confirm successful amplification. Samples with clear and positive bands were subsequently sent to first base laboratory for DNA sequencing. Finally, the obtained sequence data were edited, identified, and analyzed using bioinformatics tools to determine species identity and to evaluate the accuracy of product labeling. For this slide, I will explain the result of genomic DNA extraction for tuna samples. A total of 10 tuna samples were analyzed using 1% agarose gel electrophoresis. From this, only 5 samples showed detectable genomic DNA, which were A2, B1, B2, C1, and D1. The genomic DNA bands were observed remaining in the whales, which is expected because tuna has a very large genome size, around 755 megabases, exceeding the DNA leader range. Some samples showed smearing or missing bands, indicating possible DNA fragmentation or contamination. This could be caused by nucleus activity, heat exposure, long storage time, or technical errors during extraction. For canned tuna samples, poorer DNA quality was observed due to high temperature and pressure during the canning process, which can severely degrade the DNA. From all extracted samples, only B1 and D1 successfully amplified the CO1 gene, producing clear bands at the expected size of around 650 base pairs. The presence of sharp and distinct band indicates good DNA quality and effective primer binding. Sequencing of these PCR products produced more than 650 BP readable regions. Blast analysis showed that both samples were identified as Tunus albacares with similarity values about 99%, which indicates a highly significant match. The slight variation observed is likely due to natural intraspecific differences. Overall, these results confirm that the CL1 marker is reliable for tuna species identification. For this part, most of the samples failed to amplify during PCR even after I adjusted the handling temperature. I also tried to re-amplification using different primers like FISH2 and Tuna CR mini barcoding, but the amplification still failed. This suggests that the DNA quality or quantity was very low. For canned tuna, the failure is likely due to high heat and pressure during canning process, which caused severe DNA degradation. For fillet samples preserved with formalin, DNA cross-linking and fragmentation may affect PCR performance. In this study, only sample B1 and D1, which is fillet sample, showed clear PCR bands and produced good quality sequences. These sequences were submitted to GenBank and used for further phylogenetic analysis. For mislabeling assessment, the species identification results were compared with the product label information using GenBank and BOLD databases. In total, 10 tuna samples, including fillet and canned product, were analyzed. However, PCR amplification was only successful for 2 fillet samples, which are B1 and D1. Both samples were identified as Tunus albacares with 100% similarity in both databases. The identified species matched the label information, so no mislabeling was detected for these samples. The other samples could not be accessed because PCR amplification failed. This is the neighborhood joining phylogenetic tree constructed using CL1 gene sequences. From the results, B1 sample clustered closely with Tunus albacares sequence from China, while sample D1 clustered with yellowfin tuna from the Philippines. This indicates that both samples belong to Tunus albacares, but they show close genetic similarity to populations from different regions. This clustering pattern is likely due to the highly migratory nature of yellowfin tuna, which leads to low genetic differentiation across geographic location. In addition, Salmo-Sala was used as an outgroup to root the tree, which helped confirm that both samples are correctly grouped within the Tunus albacares clade. Next is the maximum likelihood phylogenetic tree. All Tunus albacares sequence, including sample B1 and D1 from this study, clustered closely together, indicating high genetic similarity among yellowfin tuna populations. The short internal branch lengths observed within the Tunus albacares clade suggest minimal genetic divergence and strong population connectivity, which is consistent with the migratory behavior of this species. In contrast, Tunus tinus and Tunus orientalis form separate and distinct clades, confirming clear interspecific divergence. Overall, this maximum likelihood analysis further supports the accuracy of species identification obtained in this study. In conclusion, this study shows that DNA barcoding using the CL1 gene is effective for identifying tuna species sold in Klang Valley. The results also demonstrate that molecular identification is more reliable than morphological methods, especially for processed tuna where physical features are removed. DNA barcoding helps to verify product labels, protect consumers, and support fish conservation efforts. For recommendations, further studies should include more samples, wider sampling locations, and more fresh tuna to improve amplification success rates. It is also recommended that DNA barcoding be implemented for routine seafood monitoring and that clearer labeling, including scientific names, be enforced to reduce the seafood mislabeling. Lastly, here is the references that I used in this presentation. That's all from me. Thank you for watching. Assalamualaikum. Bye.